Our protocol describes a simple in vitro system to study the effect of isolated molecules on the morphology and structure of calcium carbonate. This technique is advantageous in cases where the biomolecules at test are expensive or available in small quantities, as well as when slow calcium carbonate precipitation is required. Furthermore, it allows to probe multiple precipitation experiments under the same conditions at once.
First, prepare the control experiment. Use triple distilled water and ethanol to clean glass pieces and glassware. Use a diamond pen to cut pieces of a glass microscope slide so that they fit in a well of a 96-well plate. Place the glass pieces in a beaker with triple distilled water so that the water covers the glass slides. Sonicate in a bath sonicator for 10 minutes. Decant the water, add ethanol to cover the glass slides, and sonicate again in a bath sonicator for 10 minutes. Then, dry the slides and the glassware with a stream of nitrogen gas and place them in an air plasma cleaner for 10 minutes at 130 watts. In a fume hood, fill the wells at the corners of a 96-well plate with ammonium carbonate powder and seal the plate using aluminum foil. Cover the foil with paraffin film. Clean any residual ammonium carbonate using nitrogen gas. Place the previously cut and cleaned glass pieces into 5 different wells closest to the center. Fill each well bearing a glass piece with 100 microliters of calcium chloride solution.